Hey guys, welcome back to Abhyasa. I'm your educator Niranjan Hegde. First things first, if you're new to this channel, you have to do just press the red subscribe button and the bell icon for the notification. So we'll not waste any time, so we'll directly move to the topic. Some things have been written in the board now itself. So let's see what it is and what this topic is. The topic is transforming principle. What is transforming principle and what is of transforming principle? So transforming principle of it is DNA. So how and DNA is going to transfer its characters, transform its character in a few generations. So when it has been triggered in a such a way. So actually a person known as Frederick Griffith was studying about a pathogen known as Streptococcus pneumonia, and he observed when he observed in 1928 he was studying that particular thing. He is a medical officer, a British medical officer especially. So he observed two types of strains in Streptococcus pneumonia which causes pneumonial disease. So that is smooth S strain and rough R strain. So what was the main difference when he found out this? So smooth S strain was having a coat of polysaccharides. And it was very smooth and shiny structure it was having. And the rough strain was having no polysaccharide layer over the cell or the cell, and it was rough. It was having a rough texture over it. So he did a thing. So as he know that this causes a pneumonia, so he just ingested this particular smooth strain into a mice. He just injected into mice. So when he injected into mice, so the mice got the disease pneumonia. Okay, the mice got what? Pneumonia. It got pneumonia. Okay, so then he stated it as a virulent type. What is that? Virulent type. Virulent means which causes a viral disease. Okay, which causes not a viral disease, which causes a disease. That is virulent, right? So then rough. So he also in just the same time he ingested into mice, but it did not cause any of the virulent. That's why he just named it as non-virulent type. Non-virulent type, which doesn't causes any of the diseases. Now he learned that, so only because of the presence of the shiny coat, that is polysaccharide coat, so it acted as a virulent. So then he also again tried, so that he mixed both of the strains and ingested into the mice. So then he got a few of the mice caught the pneumonial disease because of the smooth strained ones. So mainly this was confirmed. So this was actually this is the basic experiments, and to confirm that there are two strains are there in the this thing. So but there, he did not confirmed that the DNA was the responsible for transformation of characters. So this this particular things are done by the other scientists or the biologists. So let's see what they did and how they did it. Yeah. So now the work of Griffith has been continued by the three other biologists, and they are Oswald T. Avery and Colin McLeod and Maclean McCarthy. In 1944, they started the work to prove that the DNA is having a transforming characters or principle. It has a character. So how did they prove this thing is? So they started an in vivo culture method. In vivo culture method, where there will be no usage of any animals or the mice having done. There is no in vitro. So in vivo, it is inside a laboratory or a test tube, you can say, or a petri plate or a culture media, you can say. So they started this particular mainly this experiments, so proving DNA has a transforming character. Before that, I want to tell you, before going into this experiment, I want to tell you a little bit more about Streptococcus pneumonial bacteria. The bacteria has it has two strains, right? S and R, that is smooth and rough. So the Griffith has already observed other three strains. So this in the same the other varieties he has seen, that is S1, S2, and S3 strains. Here R1, R2, and R3 strains. There were three strains have been seen in this two the substrains have been seen in this two major strains. So they used those particular substrains in this experiment. Okay, so what are those R2, R3, and they are rough strain 2, rough strain 3, rough strain 1, like that. Say same as with that of S also, smooth strain also. So what they what they did is here. So they took two strains, both the strains. One was a live R2 cells. Live R2 cells they took in a petri plate, or we can say culture media. So this is culture media, we can say. So which contains every nutrients, good maintenance of the temperature, everything has been done, and both were live R2 strain got an colony. So they got what? Colony they got, and this was live. Remember, live. And the next one was S3 strain, where it was yeast killed. So because before we only know that this is the strain which causes pneumonia, so which has been proved by the Griffith itself. But the heat kill strain, what is that? So they heated it, half killed, we can say half killed or heat killed. So they provided in the culture. They provided the culture. There was no colony has been also produced. Now they again isolated DNA from live R2 DNA, that is live R2 strain DNA has been taken out, that is isolated, and they got again they provided a culture media. Same thing happened, and there was no colony has been generated. So next they took live R2 strain, and they have mixed the culture in the culture media. They have mixed the R2 live R2 with a heat killed S3 strain, and they got the colony of S3. Why not? Then you can ask, why not live R2 they got? Because they introduced antibody for R2, which has to kill R2 strain. Only S3 got culture. So then how it has been done? So they start thinking it. So then again for that reason that they just introduced DNA. They isolated the DNA from this particular part, and they again introduced it, and they have again introduced the antibodies for R2 strain, so which has to kill R2. But again we got the S3 strain. So that means by killing even the R2 strain, the DNA of S3 strain used the materials from the cell materials from the live R2, and they have formed the S3 strains. So even now they are not convinced from this. So that's why they wanted to know, they wanted to confirm that DNA has the transformation, the live R2 strain are getting transformed into S3 strain type of cells. They wanted to confirm it. So that's why they took three different kinds of enzymes which is going to break down proteases. That means proteases is an enzyme which is going to break down. One is proteases, and the second one is RNAs, and the third one is DNAs. So I'm not going to do this again. So you can just by writing yourself, you can do it. The proteases is going to break down the proteins in a cell, right? So now they introduced this. They introduced the live R2 strain, and a DNA, the isolated DNA from S3. They with that they have now first they will introduce RNAs. They have introduced RNAs. So what has to it will destroy the RNA which is present in this particular thing. So and then even then they got the S3 strains. Then they added proteases. And again they got the S3 strains because it only breaks down the proteins, right? Then at last they added the DNAs, which is going to destroy the DNA, whole DNA. Then they got the very important thing, that is no colonies. So that confirmed that the DNA of the S3 strains, which caused the pneumonial disease, was responsible for the transformation of live R2 strain into becoming into the S3 strained type of bacteria, which has caused the pneumonial diseases. So this was proved by McCarthy, Oswald G. Avery, and McCloy in 1944. And the result was amazing, and they have been even been rewarded. So successful report has been formed on DNA as the transform. It has the transformation characters, so they can transform the characters of one cell into the other if there are two different character DNAs who are there in a cell, in a particular culture, or during the reproduction process. So I hope you understood this whole process. If any doubts will come into your mind, please comment below. I'll clear it as possible as that. So finally, I am going to tell you that like the video. If you liked this, just push the subscribe button, so I'll make more videos like this, and give a thumbs up and share to your friends, so that they will understand that there is a channel which is going to tell you about the biology thing, and they will learn. So smartly learn through this online method. So this is up for this today's video, and I'm going to tell you, let's hit the road of success together. Jai and sayonara.